Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name's Hemon from Faculty of Agriculture, Mataram University. The title of my research, Seed Stress in Fresh Growth Phases and its effect on agronomic characters and chlorophyll content of peanut genotypes. The background of my research: planting peanut is usually intercrop with perennial plantation crop as coconut, cashew, mango, or annual crop such as with corn, cassava, or okra. Intercropping system also often causes problem, especially shade stress. Shade stress affect peanut growth and productivity. One of alternative to solve this problem for increasing peanut production use tolerant genotype. Research of Hemon et al. 2018 obtained a number of F3 generation peanut lines. Each genotype had a different tolerance to shading. Growth pace of peanut plant have different response to shade stress. The objective to study the effect of shade stress given at various stages of plant growth on the agronomic characters and chlorophyll content of peanut genotype. Materials and methods. This experiment use completely random split plot design. The treatment main plot shade stress at various growth phases of peanut genotype consists of 6 treatments. The first treatment, no shade. The second, shade from planting beginning to harvest. The third, shade from planting beginning to 25 days after planting or vegetative phase. The fourth, shade from 26 to 50 days after planting or flowering to pods formation. The fifth, shade from 51 to 75 days after planting or seed feeding phase. The last treatment for shading from 25 to 100 days after planting or seed maturation to harvest. Subplot consists of peanut genotype. We use 12 genotypes, 9 genotypes of F3 generation and 3 national cultivar. And we use for shading black paranet with sunlight barrier 35%. How about results and discussion? Shading at first group phases with genotypes were not significantly interaction on agronomic traits, but show interactions on chlorophyll A and total chlorophyll. How about the plant height? The highest plant height said planting beginning to harvest. This determine the highest plant height. And how about the plant height and leaf number of peanut genotype? Genotype Takar 1, the highest plant height, and leaf number only genotype 11, higher leaf number. Table 2, the effect of seed stress given at various stages of plant growth on agronomic characters. Seed from planting beginning to harvest, the longer egg begin to flowering. This treatment egg begin to flowering the longest 4.6 days. Now how about the length of main stem segment? This treatment make longest stem segment 4.7 centimeter, and leaf area. This is treatment seed planting beginning to harvest gave the narrows leaf area 37.2 centimeter square, and plant by mesh dry weight. This treatment make the lowest dry weight by mesh dry weight, and root dry weight make the lowest also 0.2 gram, and root nodule dry weight also gave the lowest dry weight on treatment seeding from planting beginning to harvest. And how about the effect of seed stress given at various stages of plant growth and genotype on number of field pot and field pot dry weight per plant? Determine seed from planting beginning to harvest gave number of field plot only 10, the lowest number of field pot, and field pot dry weight also the lowest dry weight. And how about the genotype? Genotype C gave report dry weight the highest compare the other genotype. And how about the chlorophyll total content? This experiment show the all genotype gave the highest chlorophyll total content on the seed from planting beginning to harvest. You can see here the all genotype gave the highest chlorophyll total content. From experiment, I can conclude the first seeding at various growth phases with genotype were not significantly interaction on agronomic traits, but there were significantly interaction on total chlorophyll. The second conclusion, all peanut genotypes tend to produce the higher total chlorophyll in seed given at planting beginning to harvest. The third, seed given from planting beginning to harvest produce the lowest dry pot weight, 12.9 gram per plant, or yield decrease until 23.7%. The last conclusion, the genotypes G6 hybridization G201 with Domba produce the heaviest dry pot weight 17.2 gram per plant. This research was funded by the Ministry of Education and Culture Republic of Indonesia through the Leading Research Scheme, Higher Education Fund for Research of UNRAM Decentralization. Thank you for attention.